Okay, now that our Petri plate has solidified, we're going to open up these sterile swabs, and I'm going to do a couple different things. Let's streak my cell phone. What do you think? There's bacteria on the cell phone? Okay, and then streak it on the Petri plate in a zigzag type of pattern. Okay, so any bacteria on the cell phone will grow on this nutrient agar. The other thing I'm going to do is swab my hand. Let's get between the fingers. Let's get all over everything. Hey, what do you think? Bacteria gonna grow? I think so. All right. And I'm going to label these and put them in the incubator. Okay, I'm gonna put these in an incubator that is set at human body temperature, because these bacteria came from the human body. I'm putting them upside down so that the condensation that's on the lid doesn't drip onto the surface. So we're gonna give these 24 hours and we'll see what grows on them tomorrow. Okay, let's check on our Petri plates. All right, look at this. Got some nasty stuff growing on there, some nasty stuff growing on there. So now, let's look at these a little closer. Okay, to examine these, we're going to use the Quebec colony counter, and it's got a magnifying lens. We can put this down here, and you can see that I've got bacterial colonies all over the place on my hands, and from my cell phone, I've got some more distinct colonies. So let's go back to the table. Okay, we saw the Quebec colony counter. Now we want to describe the colony. So let's look at these Petri plates. Okay, so these are irregular white. These, you see this one's slightly yellow, and we got a whole bunch of little tiny dots. I think because these colonies are bigger, I'm going to sample this one. And in particular, let's look at this one. Okay, we're going to choose this bacterial colony, and first we want to describe its color. So the color, let's say white. Now we need to measure it. Okay, we're going to measure in millimeters, and we'll measure the length is just about 20 millimeters, and the width is maybe about 10. Now this isn't rocket science, so we're going to say 20 millimeters by 10 millimeters. The next thing we need to choose is the form, and they can be either tiny dots, perfectly circular, or irregular. What do you think? I'm going to go with irregular. Okay, and then finally the margin, that's the edge. Is it perfectly smooth? Is it a little wavy, what we call undulate? Or is it big lobes and extensions? I'm going to go with undulate. All right, so now what we're going to do is sample from this Petri plate, and we want to grow a whole bunch more, so we're going to put some of the bacteria into an agar slant. We're going to use an agar slant, which is solidified at an angle, and we can inoculate it on the surface. Now the question is, how do we go from the Petri plate to the agar slant? Well, we're not going to use our hands, we're going to use a transfer loop. I know you can't see it well, but at the end of that wire there's a loop, and we're going to heat up that wire to sterilize it. So we've got our Bunsen burner, turn gas on, and there's our flame. So you just hold the wire in until it turns bright orange. Okay, you don't have to sterilize the handle. You especially want that end to be nice and orange. Okay, now that's sterile. We let it cool. Okay, we don't want to blow on it because there could be bacteria on our breath. And now we take our Petri plate and we're just going to sample, just take a little bit, okay, just a teeny tiny amount. Open up our agar slant and reach inside there and zigzag it Zigzag it up and down. And what we're doing is we're trying to grow more of that bacteria for further testing. You'll also notice I just sterilized this before I set it down because we don't want bacteria getting on the table. So now I'm going to label this and put it in the incubator. Now, the other thing we need to do is a gram stain. So we're going to take more from there, put it on a slide, and do a gram stain as written in the PDF. Okay, here's my unknown bacteria. Notice that they are rod-shaped and they're kind of stuck together like strands of hair. Those are called bacillus shape. And they're purple, very dark purple. So we're going to say gram-positive bacilli. Okay, so my bacteria were very dark, almost black, rod-shaped. Some of them were kind of close to each other like this. Okay, so now what would those be? They're gram-positive. Are they staphylococci? No. Streptococci? No. Diplococci? Tetrads? Wait, bacilli. So these are gram-positive bacilli. You should know all of these different shapes of bacteria. But now we go to this table. We say the bacteria source was the phone. It was gram-positive. And the cell morphology is bacilli. All right.